Hello, my name is Tristan, and today we will be talking about the anatomy of a pipette. Just kidding. Instead, we will be time traveling to this region of India about 66 million years ago. And hold on. We'll be time traveling to this region of India about 66 million years ago. Imagine a deadly landscape full of volcanic activity with molten rock exploding from the ground. Eruptions happening every couple minutes due to the extreme pressure exerted below the Earth's crust. From each eruption, deadly gases emerge and fill the atmosphere, causing intense global warming. Because of this, the oceans are changing and suffering, increasing in acidity and losing oxygen. This was a dark time in the history of life, literally, because as all these things were happening, a giant asteroid struck the Earth, and it was 1 billion times more powerful than the atomic bombings of Hiroshima and Nagasaki. This event was known as the fifth mass extinction, commonly known as the Cretaceous extinction, which happened roughly 66 million years ago. About 76% of species were lost, including the dinosaurs. 66 million years later, the world seemed to return back to normal. Those who survived speciated over time and assumed their niches all over the Earth. That is, until humans arrived. We are in the midst of the Earth's sixth mass extinction. Much like the dodo bird, various animals you know and love are predicted to go extinct, such as rhinos, monkeys, lemurs, giraffes, big cats like lions and leopards, marine mammals, sharks, whales, seals, countless species of amphibians like frogs, and it goes beyond animals too, but there is just no time to list everything. To most people, a mass extinction looks and feels like a huge loss, but it isn't clear how we should address it. Is it our fault? Is it really that bad? The short answer is yes. The long answer is a bit trickier, and it has to do with a lot of various different factors. Most of them are human caused. If you are curious about what is happening with the sixth extinction, take a look at this book by Elizabeth Colbert. Or if you want a slightly shorter read, this article in Nature sums it up fairly well. So why am I talking about this? What is the big deal with mass extinctions and extinctions in general? Well, life on Earth is a story, and we vaguely understand it. The history of life is full of unique circumstances that likely changed the course of what it became now. Remember the meteor from before? That circumstance changed everything, and it killed most of the big animals on Earth. And it sparked the rise of mammals and flowering plants. Stop thinking of life as a thing that just exists, and it is the way it is. Ask yourself, where did eukaryotes come from? Was it inevitable, or was it just a circumstance? Did their chromosomes become linear because of random double-stranded breaks in their ancestors' circular chromosomes? Or was it an inevitable thing that eventually became beneficial for survival? There are several keystone transitions throughout the history of life, such as the evolution of multicellularity. That transition between single cells and a multicellular creature, like the origin of animals, was that inevitable? Or do cells naturally drive to live together in a colony because living in a society is beneficial? Let's try a thought experiment. Imagine a pond called the primordial soup pond with a colony of cells swimming around, having a blast, absorbing nutrients from the environment, doing their thing. A tornado comes and scoops out a group of cells and transfers it to another pond far away. Over time, this group of cells likely has speciated and changed a lot. Of course, if something like this happened in the past, we probably will never know. It is a weirdly specific story, but I like to think about it as a thought experiment of the unique situations that might have changed the course of life. It is much like how we study human history. There are plenty of interesting stories of the past, like the Great Wars or personal phenomena, like the execution of Socrates. But here, it is no doubt that there are many things that we just do not know around the time of Socrates. There may be may have been many fantastic works or stories rich in detail and intriguing, or personal stories throughout human history that we just do not know about because they haven't been recorded that well. There just isn't any evidence of it. That is why history is so fascinating. It is intriguing to find new evidence of something that no one ever knew existed. Now think about it in the history of life. Think about it in the mind of a biologist. Think about that. It's billions of years. That is so much history gone unaccounted for, and only a tiny, tiny percentage of it remains as evidence of its prior existence. And only that percentage is due to some characteristic of life that can produce a solid substrate that doesn't degrade over time as much as organic matter does. Bones make good fossils, but bones only arose in the vertebrate lineage only millions of years ago, not even over billions of years. Think about the parts of life that we just don't have evidence for, and all of the lineages that may have gone extinct, and there's only a very small lineage that exists right now that we think is so diverse, so insanely broad, crazy, and beautiful. Well, it is, but there's just so many things unaccounted for in life, because there may be no evidence of it, no leftovers. But there are some leftovers in our own cells and some organisms that are alive now that can tell us about the past, because that's the way life works. There are some hints at what it could have been like. Studying life is, there's some hints at what it could have been like, but there's just no clarity. And that's why studying life is so interesting, but also very frustrating as well. Life is mysterious and extremely complex. It is full of bewildering and fascinating puzzles. Figuring out just one small mystery will give a biologist a high. There are mysteries at all levels of life, at the cellular and molecular level to understanding the behavior of cells or other organisms, understanding the crazy interactions between living things, at the biosphere level, that the Earth is just one gigantic system that goes in and out of balance. And we are just starting to understand the diversity of life on Earth now. A traditional biologist may spend their whole career just figuring out one little facet of life because it is so interesting, but it's also very difficult to study. So you spend your whole life studying one little thing. Being a biologist is kind of like a double-edged sword. What we find most interesting also makes our jobs most difficult. We love solving mysteries, but some mysteries can't be solved completely, and other mysteries we may just never know about. What makes me really sad is that about 4 out of 4 billion species to evolve in the past 3.5 billion years, about 99% are gone. Extinctions are not uncommon. And it may surprise you to know that we cannot study organisms that have gone extinct. We have some evidence of prior forms of life, like fossils and DNA, but the answers are usually vague and unclear, devoid of detail. We only have a general understanding of big living concepts that happened in life's really long history. However, there are plenty of mysteries to solve right now. And if you are a biologist and you want to be a biologist, there's plenty of things to do, and there's plenty of interesting things to try to figure out. It's really exciting. There's more than enough evidence to look at and experiment with. So that brings me to an important topic. How do we study life? Well, we look at it. We look at life. But it is slightly more complicated than that. Living things are difficult to study, especially at the cellular or DNA level. Most of what we know about living things comes from something called model organisms. These guys right here. These are really easy to grow in the lab and study, like the plant Arabidopsis right here, which has been extremely important in understanding how plants work in their genetics or other qualities like their immune systems. However, using the few models we have as representatives of life could be a problem. If we only study Arabidopsis, it doesn't necessarily tell us about every other plant. because Arabidopsis is a flowering plant. It's vastly different from ferns and mosses. So it can't tell us about everything, but it has been extremely useful in telling us about plants overall. Only studying the few model organisms we have will give us a very skewed perspective on life. Let me repeat that because it's very, very important as a concept. Using the few model organisms we have will only tell us and give us a very skewed perspective on life. The foundational level of knowledge in a biology textbook is useful. It's good as a beginning reference to understand how life works, but be aware that it cannot apply to every living thing. There is always an exception to the basic knowledge of biology in every single field. So we must look at the foundational principles in a specific way. What model organisms did that knowledge come from? And does it apply to every living thing, or is it unique just to that model? There are plenty of mysteries in life that we need new models to solve them. So now let's talk about the problem of extinction. Like I said earlier, we cannot study extinct organisms. Simply, they're just usually are not here, and the evidence that they might leave behind is not that easy to study. If fossils, like bone fossils, are just not particularly that useful, you know that that organism existed, but may not tell you much beyond that, how they worked and other things. Yeah, especially if they do not leave evidence behind of their prior existence. We know now that we are in the midst of the Earth's 6th mass extinction, which is caused by humans. So what am I doing about it? Like, what am I doing about the problem of extinction? Most people will address the problem of extinction by fixing environmental issues. There are many problems caused primarily by humans that need to be addressed, such as habitat loss, the introduction of invasive species and pathogens, the overexploitation of wildlife, pollution, climate change, and carbon emissions in general. These are just some of the qualities that are very devastating to life on Earth right now, and it's causing a lot of extinctions. These are fairly big issues, but I want to address the problem in a different way. I choose to capture as many new forms of life as I can before they go extinct. That's how I am addressing the problem. And it's useful because other biologists can use new forms of life that are discovered as model organisms to solve some truly unique mysteries that they bring up. Some models, like I'll talk about in a bit, have really changed the way we think about how life is structured. And this is what I'll be focusing on for the rest of my career. So yeah. So I'm interested in hunting novel microbes. And there's a really good reason for this, because microbes are amazing, just in their own right. To show you why, I have to tell you what microbes are in the grand scheme of life. So what is life? Well, life are cells and cells are life. This is a fairly simplistic model, but life is a physical piece of information that leads to its own replication. Living things reproduce. And the way it does this is that the information codes for a machine. This is the information, we call this DNA, and this is the machine, a protein, that will lead to replicating the information. So the information codes for a machine that grabs onto the information and copies it as such. This all happens in a cell, and when it replicates itself, the two strands of information get split into two different cells, which are pretty much the same. But they can also be slightly different. We know now that this information is DNA. This may not always have been, though. May not always have been DNA, the information. These two cells will go on to 4, then to 8, then to 16. Life persists by replicating itself. That is the fundamental principle of life. If it couldn't replicate itself, it will never have existed. Everything a cell does is to persist in the environment, to survive, and to reproduce or replicate itself. They need energy to maintain their body. They need to move around and escape danger. These are just some of the things a cell will do to survive. So yeah, they need, they use proteins to facilitate all of this. These are the tiny machines that work and run everything in the cell. The information will code for these machines, these proteins that will run all of these different things. And they facilitate pretty much everything. The reason I'm telling you this is because I argue that life is cellular based by necessity. I know it seems like a concept that I shouldn't have to argue, but most people don't think about why are living things cellular? Like why do we have cells? What's the point of that? Well, it comes down to that it seems that it needs a compartment to contain everything. A cell is just a compartment, a space to hold all of the complex machines and information that makes life what it is. I can prove this by looking at non-cellular life. As you can see, the machine will, it'll float away. So everything will just float away if you're not contained in a small compartment. Everything will float away, and it probably will never get to reproduce. Living things need to be contained, and that is why cells exist. That's why living things are cellular based. Here's a fantastic example of a cell. It's beautiful, it's very complex. You can see how intricate it is, and it's splitting, getting together, and yeah, it's really cool. Now, we live in a microbial world. Most of life, the microbes, are single cellular, or at least we can't see them that easily. But just because you don't get to see them every day on your evening stroll does not mean that they aren't charismatic, because microbes are charismatic. Sometimes you just need a microscope to see their charisma. Microbes were the first rulers of the earth. They've existed long before animals or plants. If we look at this timeline here, we can see the origin of life around here. And over this time, we think that these are most likely simple-ish single cells back then. They kind of stayed where they were. They reproduced a little. They absorbed nutrients from the environment. Not too much going on, but they were living. They were replicating and reproducing. And then something happened across a billion-year stretch. So these living things lived for about, quite simply, a few billion years, and then something else happened. A small group of these little cells turned into the eukaryotes. The eukaryotes have emerged, and these are really crazy. They're still single cellular and they're microbial, but they're profoundly complex, larger, and I think they're a little bit more interesting. Every now and then, we see throughout the history of life, these are the microbes that emerged at the beginning. That's just how life is. Living things are cells, like I said before. These are the microbes. They lived for billions of years before a single multicellular creature emerged. Every now and then, some multicellular creatures emerged, and we know these as algae and plants over here, and also probably your favorite, the animals, about right here. Only 600 million years ago, that's when the animals sprung up. Now let's look at the tree of life for a real quick. Boop, boop. Let's go back here. Oh gosh. Yeah, here we are. Here's the tree of life. Just to show you a broad aspect of what living thing is, like how diverse is life itself. These are the bacteria. These are the insanely diverse. Like bacteria were among the first organisms to appear on Earth, and they have existed from the beginning to all the way till now, and they are insanely diverse. And there's so many different qualities of bacteria, and there's different types of bacteria that are just crazy in how they work. These red dots right here show us all the lineages that we don't have any isolated representatives, which means that they haven't been studied in the lab. These bacteria, most of them, really, this whole phyla right here, just hasn't been studied in the lab yet, but we do know that they exist. Same with the archaea. Archaea are really crazy in their own right. And then this little branch here is called the eukaryotes. These are those big cells we talked about earlier. And we zoom in on the eukaryotes, we see this nice huge tree of life. And what we see is all of these lineages here, these are microbial. And if we look at this tiny little lineage right here, we zoom in, I can't zoom in any further, but this tiny little lineage right here, metazoa, that is the animals, all of the animals that exist, everything you see every day, the animals, insects, birds, you name it. You are here and all of the animals are right here in this tiny lineage. Then the plants, every plant you see every day is right here located at this lineage, and every other lineage besides fungi are right there. Every other lineage are microbes, either single cells or maybe algae and other things. So most of these are just microbes that you do not get to see every day. And that's most of life. So most of life, you don't get to see every day. You have to get a microscope or something. All right. Next. Yes. So every multicellular creature evolved from a single-celled ancestor as such. The only thing that makes a multicellular organism is that the cells stay physically attached together. And it's a really long transition to become multicellular, but every multicellular organism does have a single-celled microbial ancestor. But being multicellular is very useful. You can divide labor up between groups of cells. Say this group is dedicated to eating and this group is dedicated to moving or reproducing, just as an example. So being multicellular is pretty nice. Microbes are full of oddities, the exceptions to the current foundations of biology. Microbes are everywhere. The closest relatives of the first cells that live on today, we call them bacteria, that we talked about earlier. Bacteria and the single-celled eukaryotes are the most diverse forms of life. We saw that in our tree of life. They are everywhere. They're in our gut, they're on our skin, they are in every other organism you can think of, everything you see every day. A bird has tons of microbes in it and on it. Grass forms association with microbes, and they need these associations to really live. So every other organism has microbes either inside it or on it, or even inside their own cells. And they're also everywhere else. They can be inside of a single eukaryotic cell. You can find about 1,000 different types of bacteria that are intracellular bacteria, which is crazy. And even other eukaryotes can be inside of that host. And that's just how life works. Living things can live inside of each other. It's very, very common. Living, like cells will live inside of other cells, which is crazy to me. It's really, really crazy. Microbes also colonized most of every possible place on Earth, from the deepest sea to the soil and to the coldest regions like Antarctica, or these lovely hot springs you can find in Yellowstone. What surprised me the most in my studies is that new microbes are discovered almost every single day. There is plenty of undiscovered life out there, and some of it is absolutely bizarre. But there are also not that many researchers who go and look for new forms of microbial life. Most biologists do not become microbiologists, and they don't particularly study, and even most microbiologists don't study microbes that don't affect humans as much or directly. So there's many things to do if you want to become a biologist. There's plenty of things to do, actually. Meet Jana Iglet, a personal hero of mine. She was previously a PhD student at Alistair Simpson's lab, who is a fantastic protostologist at Dalhousie University in Halifax, Canada. Jana and Alistair discovered this particular microbe in the forest of Halifax. And it is they looked at it, and it is a predator of other microbes, and they said it can be fairly aggressive on a small level. Alistair said this discovery literally redraws our branch of the tree of life at one of its deepest points. They essentially found a new kingdom of life. And this was 3 years ago, in 2018, which blows my mind. Like finding a new kingdom of life, that's insane. And that was so recent too. What else could be out there? We do not know. This microbe formed a new kingdom of life, and Jana worked to culture it in the lab so that they can study it as a model organism. So I'm really excited to see what they find in the coming years. Now we move to part 3, my actual thesis presentation. Apparently, I'm required to actually talk about my research from the last 4 months. So here we go. When I started this project, I wanted to do one thing, to hunt novel microbes. I hope I gave you enough reasons as to why I wanted to do this. It turns out that I did not get to finish the project I started last spring and last year for reasons I'm sure you guys are aware of. To keep it short, I'm not going to talk about which microbes I was hunting because I haven't really found them yet. I've only done like maybe 1 or 2 experiments, and it takes a little while to find what you're looking for, so to speak. So far, I've only completed like 2 isolation experiments. If you guys are interested in what exactly I was hunting, feel free to ask me when we start questions. So if you're curious about what I was hunting, it's a long story, but you can ask me later. So here's the technical title of my thesis: An unexpected isolation of antibiotic-resistant Vibrio Harvey Clade bacteria from the guts of Galveston Bay bloodworms. To simplify it, I basically found a weird bacterium that lived inside of bloodworms, which is a worm that lives in the sands along the coast. So if you go to the beach, they're probably living underneath your feet. This bacteria was antibiotic resistant and very likely pathogenic. It also might be a new strain or species, but it's too soon to tell, and more research is needed to confirm what it is. Here is the worm that was my model for this experiment, this specific experiment. I called it a bloodworm because it looks like blood, but it's really a marine polychaete, a bristleworm in the family Orbinidae. And this is just what it looks like, a little video. There it is moving around. Very nice. And this is the phylogeny, if you're curious. So I went to Galveston. So here's the world right here. Here is the US. Here is Houston right here. And if we zoom in right here, this is Galveston. This is Galveston Island. Right here is Galveston Bay. This is the West Bay. Right here is San Luis Pass. And right here, where this little red thing is where I isolated the worms. That's where I found the worms hidden in the sand. So I dug them up about maybe a half a foot into the sand, or maybe 2 centimeters to 2 inches actually. And I found them there. So I got about 15 worms I collected, and then I took them to the lab that day. Then I cut them open, took out their intestines, and I made a slurry from their intestines and their internal organs so that I could isolate the microbes inside of them. Then I plated the contents of their intestines on a medium they can grow on, but I actually used antibiotics, so bacteria could not grow. It turns out that one bacteria did end up growing, however. So I was kind of intrigued. Why did this bacteria grow, and what is it exactly? Because I found 5 different colonies of the same bacteria that seem to have just came out of they did come out of the guts of the worm. So there's also a yeast, but I didn't look at that too much, but I focused on these bacteria right here. So that's what we're going to look at. So this is what the yeast looks like. I ignored it, but it looks like this when I stained it. Here is the bacteria. So this is a gram-negative bacteria. When I stained it, it was gram-negative, and as we'll see later, that makes a lot of sense. There's about 5 colonies of it, and here is what the colony looked like. It's pretty weird. It has a very unique morphology, and I haven't seen a morphology like this before. And when I looked in the literature, I haven't really seen any other colonies that look like this, and I couldn't find anything. So I called it Metro, short for metropolis, like a little city, you know? And there's some more microscopy that you can see. It's a rod-shaped bacteria that seems to be in like a glycocalyx-like substance, so like a slime layer or something like that it's producing. And this is a professionally drawn cross-section of the colony. It would probably look a little bit like this. This is called an elevation pattern. When you're looking at bacterial colonies, you kind of see what it looks like on the side. So that's what that looks like. So I took upon identifying what this bacteria was because I was just curious. So to identify it, we sent off 3 of the colonies to get sequenced at GeneWiz, which is, if you don't know, a biotech company which will basically do a lot of your research for you. And I got back their 16S ribosomal DNA sequences. And ribosomal RNA itself is really good because everything has it. Everything in life has it because ribosomes are needed in life to synthesize proteins. If you're a bio student, you probably know that. So every single cell, every bacteria, eukaryote, animal, plant, whatever, everything will have ribosomes. And every ribosome is related because it's very, very well conserved amongst the tree of life. So biologists will use ribosomal RNA, the gene itself, to compare between all other forms of life to generate a nice tree of life showing how related different organisms are. So in bacteria especially, you use the 16S ribosomal RNA gene, and that helps you determine where your bacteria fits relative to all of the other bacteria in the world. So yeah, this is sort of what the sequence looks like. We're not going to go too much in the methods, but I processed all of the sequences. And if you're curious about how I did that, you can ask me later. For some reason, if you're curious about that. Here is what I made. This is a little tree. So to navigate this, we're going to look over here. So right here is Metro. Metro, these are the bacteria that were sequenced. And everything else around it are relatives of Metro. So these are very well related. Everything else away from it is not as well related. And these right here are other groups of bacteria that are just huge groups of bacteria, like cyanobacteria and some other things you may know and love, like E. coli is over here, is known as a gamma proteobacteria, and agrobacterium and rickets, things like that, chlamydia, and all kinds of stuff. But this in cyan is Vibrio, just the genus Vibrio. So these are all of the Vibrio genus of bacteria that are sequenced in the world. And I compared it against my strain of bacteria. And what we find is that what this bacteria is a Vibrio species. Specifically, it's in the Vibrio Harvae clade. And I know that because it is very well related between all of these Vibrio Harvae clade members. So there's that. So what does that mean? Why does that matter? Well, this is another tree of life. This is showing the genetic distance between everything. So how well related everything is shown in a map. So all these Vibrios are very well related. That makes sense because they're the same genus. Could be different species though. Everything else is very distantly related. Like these cyanobacteria, these are a group in their own because they're cyanobacteria. So what could it be? What could this mysterious gram-negative bacterium be besides just a Vibrio species? What could it do? What's the purpose of it? Well, the answer may not be very nice. This Vibrio is very related to a species that produces tetrodotoxin and pufferfish, which if you don't know is a very dangerous toxin. Very toxic. Another relative of this bacteria is flesh-eating bacteria that we hear about almost every summer, especially in Galveston area. Necrotizing fasciitis is what it causes. Very bad disease. Another relative is known to cause mass death in clams and crabs, which can be pretty devastating to the seafood industry. And also another relative can cause seafood related sicknesses. Like if you've ever ate a raw oyster and you've gotten sick from that, that was probably due to one of these Vibrio bacteria, because Vibrios are very much famous for being pathogenic and dangerous. So this is kind of a warning sign for Galveston. This probably shows us that this bacteria that I isolated from the worms is probably very dangerous, and it's also antibiotic resistant, which is not good, probably very, very much not good. And we know that antibiotic resistance is rising in the world amongst many bacteria, and there's various reasons for this, but it's not a good thing because how are you going to treat them if everything is resistant? Further research is needed to assess this appearance of the bacterium and the bay, and we need a little bit more research to figure out what exactly it is. So yeah. Also, Metro died over spring break. So all these bacteria died right after I sent them off for sequencing. So I couldn't work with them any further. So I couldn't do any more experiments that would have been interesting to pursue, but I did get their sequences and I could identify them pretty well. So they were unrecoverable. I really did try growing them again, but it just didn't work. The experiment could be reproduced if we pursued it further. So if someone wanted to pursue it further, you could definitely redo the experiments with the bloodworms, because it's very likely that you might be able to find them again if you use antibiotics, because they're pretty prevalent in the sands. So with that, I'm going to kind of go past this project now and take a look at future directions. I'm not going to continue this specific project because I'm graduating. If you want to read the thesis for some reason, I can get you a copy. Just send me an email right here at hendersontc@hvu.edu if you want to read it.